Hello, here is a short video about precision tumor recognition by T cells with combinatorial antigen sensing circuits, where we show how we can target tumors without causing any harm to normal tissues in the body. To begin with, T cell is a type of lymphocyte which constitutes the immune system in the human body. T cells are characterized by what's called the T cell receptor that can recognize an antigen infecting the body. This antigen can be any tumor, for example, a cancer cell. So the complementarity between the receptor and the antigen enhances the binding of these two molecules, hence allowing the T cell to kill the cancer cell. T cell can be synthesized in the way that its receptor, known as chimeric antigen receptor, CAR, or T cell receptor, can target a specific antigen. However, targeting single antigen can be harmful for normal tissues, described as bystander tissues, leading to life-threatening. So this paper shows that a combinatorial T-cell circuit can be engineered where a synthetic Notch receptor that targets the first antigen activates the expression of the T-cell receptor that targets a second antigen and induces its immune function. So this process requires the presence of two antigens at the same time to be targeted by the two receptors on the same T-cell. Hence, it was demonstrated that these engineered T cells specifically bind to combinatorial antigen without causing any harm to the bystander tissues. So, as shown in this slide, the presence of antigen A activates the SYNOCH receptor, which in its turn triggers the release of transcriptional factor marked by TF. So, the TF can now activate the expression of the gene that encodes for CAR or T cell receptor that has a domain that can bind antigen B, and indeed, the T cell is activated by the binding of both antigens that can be cleared now by the T cell. So the activation of these engineered T cells require the presence of two antigens. In other terms, the presence of antigen A only or antigen B only cannot trigger the activation of the T cell, and as a result, these T cells will not affect the normal tissues and will not recognize single antigens. To make sure that the synthesis of these T cells is effective, it has been tested on Jurkat cells, which are a form of human T cells that can target different tumor antigens. CD19 and mesothelin are two antigens that can be recognized by Jurkat cells and which were used in this experiment. So the Jurkat cells were engineered in a way that they contain alpha CD19 SYNOCH receptor that can bind CD19 antigen and alpha mesothelin CAR gene that can be transcribed into alpha mesothelin T cell receptor that can recognize mesothelin antigen. When the engineered T cell was cultured with mesothelin only, no activation of the alpha mesothelin gene has been observed. However, in the presence of dual antigen that has mesothelin and CD19 with the Jurkat T cell, it has been observed that the binding of CD19 to the SYNOCH CD19 receptor triggered the transcription of the alpha mesothelin gene. So the activation of transcription leads to the formation of alpha mesothelin CAR that can bind mesothelin. The binding of both antigens at the same time leads to the activation of the T cell and the release of CD69 and interleukin-2 cytokines that regulate the body to fight against this dual antigen and clear this dual antigen only without causing any harm to bystander tissues. So the SYNOCH receptor has successfully driven the CAR expression to kill the antigen. The study has also tested if the SYNOCH receptors will drive the human primary T cells, CD4+ and CD8+, to kill target cells. Alpha GFP SYNOCH receptor was used in this case that when bound to GFP antigen can activate the expression of alpha CD19 CAR gene. These T cells were cultured with K562 target cells that can express both CD19 and GFP antigens. It has been observed that both forms of the primary T cells, CD4+ and CD8+, were only activated when exposed to both antigens at the same time. Also, the same procedure was followed using two other SYNOCH CAR combination. The same results were observed also here, where both CD4+ and CD8+ required a combinatorial antigen to be activated. So, putting everything together, the use of synapse receptor to regulate the expression of the T cell receptor to target antigens has shown a big success and a safety for normal tissues, which will help now to target a wide range of tumors without causing harm to the normal tissue with very little side effects on the human body. Thanks for watching.